hydrogenetic receptor with novel therapeutic perspectives. Let's hope I didn't butcher that too much, Krzysztof. Okay, thank you.
Yes, thank you very much. And I'm really happy that I can make this speech today. And before I start my presentation, I would like to say that most of the speakers are presenting outline with saying what they are going to talk about. And the more recent trend is also that they are talking what they are not going to talk about. So I would like to make a claim that I will not talk about chromatography at all, even though this is a word for chromatographic fields for Dr. Weiners. I'm medicinal I started my career as an analytical chemist, and I'm very happy that Earth's made some comments on my previous results on this, but I would like to present you the purely medicinal chemistry project. So starting with some background. Phenotral is a compound which is a selective agonist of the beta-2 adrenergic receptor, and it was used, and it's in some countries this is still in use as a treatment for asthma and bronchitis. It was introduced in the 1986 as a racemic mixture of RR and SS forms, and in many countries, including USA, it is withdrawn due to severe adverse effects. And the title of this reference here tells quite a lot about the reason. So patients which are using this medication, or which were actually misusing or overusing, got severe cardiac problems, which could eventually end up with a cardiac arrest and a death of the person. And having this into account, the scientists in the National Institutes on Aging started up a new project where they combine selective optimization of side effects with a chiral switch approach to develop a new highly selective agonist for beta-2 adrenergic receptor, which in combination with beta-1 selective antagonist can be a promising strategy for a therapy of some cardiac problems and congestive heart failure in particular. And in this project, number of new phenotral derivatives were synthesized and tested for efficacy, and I'm going to show the progress of this project. And the idea obviously is to show the project and to show the Dr. Weiners' life, which goes beyond analytical chemistry and chromatography. Okay, so this is Fenoterol. It has two chiral centers, so there is 4 different stereoisomers, and indeed binding to beta-2 adrenergic receptor is very different for each of the stereoisomers. This is the most active, this is the least active, and when we talk about selectivity between beta-1 and beta-2 subtype of the receptors, we can also see that there is quite a big difference. It's for RR phenotral, this is about 44 times lower affinity to beta-1 than to beta-2. That was the first stage of the project, and these 4 stereoisomers were synthesized, and actually, so I will mention chromatography. It were resolved on chiral stationary phases, and I think Carol will also can refer to this. Okay, and at the first stage we developed some 3D QSAR models, and we performed some structure design, which I would like to briefly present to you. So in the first step of this project, 26 structures were synthesized of those compounds. They were 7 different derivatives modified at these places, and each of them were synthesized in every possible stereo configuration. So we finally had the 26 structures, and all of the compounds were characterized for binding to beta-1 and beta-2 receptor, and we built 3D QSAR models to explain both stereo and regio-specific aspects of interaction with protein target. The approach we selected was comparative molecular field analysis. It was important because all the structures were the binding affinity highly depends on stereo configuration. So we had to use a three-dimensional type of models. This is a model. It can be nicely interpreted in a way to explain why first chiral center is important, why the second chiral center is important, how or what type of interactions are responsible for stereo selectivity in this case. But the most important part, the model has a very high statistics significance, and we decided to use this model to design some new structures. So in step 2, the CONFA model was used to predict pKa values for compounds which we didn't actually have. We just draw on a piece of paper and then draw into a computer, put into the model, and predict the possible pKa values here. And these are predicted numbers for different stereoisomers, for different possible structures. And after that, we decided to synthesize 6 of these structures and determine experimental value of pKa. And as we can see, there is a quite good correlation or agreement between computationally predicted number and experimentally determined value. The R-square press, which is a correlation between this column and this column, is 0.73. So it's quite high. And there are a couple of other steps, and due to the time constraint, I will not present them. But in the whole process, we developed a couple of lead compounds, and one of them is RR4-methoxy-1 naphthyl derivatives with this structure, which has a remarkable subtype selectivity. The binding affinity to beta-1 is almost 600 higher than the binding affinity to the beta-1 subtype of the receptor. Another lead compound we consider here is ethyl derivative, which also have a very high subtype selectivity, but it appeared also important in later studies on some tumor cells, which I'm going to present later on. And another lead compound we consider here is RR-methoxyfenoterol. The compound appeared so important that we decided to synthesize the compound in a tritiated form. So use it as a marker in radioligand displacement studies to study phenol derivatives and also many other compounds bounding to the receptor. The important is why this is important, because it looks to us that the new compound probes the binding affinity of ligands to the active form of the receptor. As you can see, that's if we test agonists and also phenol, we can and all the phenol derivatives, we can see that binding affinity determined with new radio-labeled ligand is much higher than it was determined by other means, while testing antagonists, this is exactly the opposite. So we could show that now binding affinity depends on the radioligand we used, and also depends on whether the compound is agonist or antagonist. Okay, in the second part of this study, we decided to determine thermodynamics of binding. And we did the Van Hoff plots for binding affinities for the same experiments as we did earlier, but the only difference is that now we are testing how binding affinities changes with temperature. And here is the data. This is a RR Fenoterol, which indeed has the highest affinity, but its affinity does not depend on temperature, actually. Does not significantly depend. When we tested RS Fenoterol, we can see that binding affinity is increasing with increase of temperature, but the most stunning thing we observed here is completely the opposite trend for two other stereoisomers. And it was something really unusual and not frequent. Actually, that's to my knowledge never shown that binding affinity or thermodynamics of binding is completely reversed for one enantiomer than in comparison to the other. When we interpreted it, we could determine that when we interpreted Van Hoff plots, we could determine that RR and RS Fenoterol has a purely entropy-driven binding. These are two compounds here, while two other stereoisomers, SS and SR Fenoterol, they show a predominantly enthalpy-driven binding. So completely different mechanism and completely different mode of binding, which were determined in the studying of thermodynamics in this system and in the formation of a complex between a target protein and different stereoisomer of phenol. That was the results on binding affinities. Now I would like to show a couple of slides on functional efficacies. Actually, this is a table, kind of master table, which shows comparison of, in two first columns, binding affinities determined with two different radio-labeled ligands, and also functional efficacies, like induced stimulation of cyclic AMP accumulation, cardiomyocyte contractility, and one additional experiment, which was inhibition of mitogenesis on the one of the astrocytoma cells. And in each column, I mark in red top 5 or top 4 compounds, and as you can see, in some cases they are more or less showing the highest affinity in the set, like for example RR Fenoterol, but there are also differences. And one of the difference I wanted to point your attention to is RR ethyl Fenoterol. I already talked about this compound, which has a moderate affinity, not very high, moderate induced cyclic AMP accumulation properties, but very high activity on inhibition of growing of the tumor cells, brain tumor cells. But the most important part is it does not show almost completely the activity on the cardiomyocyte contractility. So we should expect that using this compound in a patient does not create cardiac problems, or should create much less or lower problems than when we use many other compounds in this set. So that's why we consider this compound as quite interesting, because we could perhaps use it as a possible treatment for brain tumor cells without significant problems from a cardiac system or a cardiovascular system. Okay, and now I'd like to show a couple of slides on something which is really interesting, but it needs some additional introduction. So first, Beta-2 adrenergic receptor belongs to the family of G protein coupled receptors, which means that this protein couples to G proteins. And beta-2 receptor is one of a few which is known to couple to more than one G proteins, and in this case it interacts both with GS and GI protein. So upon agonist binding activates both GS and G proteins and activates different intracellular events. On this, we can separate these two events by using a pertussis toxin, which blocks a GA-mediated pathway, and in a cardiomyocyte contractility assay, when we use this type of experiments, we can see that where the experiment is pre-treated with PTX, the dose response curve is shifted to the left. So it shows like a compound is more effective than in comparison to the control experiments without PTX. And this is something which we could observe is in most of the experiments for most of the beta-2 adrenergic agonists, including such agonists like SR Fenoterol or SR methoxyfenoterol. What we could observe, though, is that some derivatives, and only in specific stereo configuration, activate selectively GS protein. We could see it by comparison of a red curve and black curve, when in case of these two compounds, there is no significant difference between those two curves. So it shows that PTX is not affecting the experiments, which shows that a receptor, upon bounding of this specific ligands, activates to the form which selectively couple to G protein only. That was some preliminary first experiments which draw our attention to something which is very interesting and very hot in the, I would say, in the current pharmacology, which is called the biased agonism, which means that different agonists can activate the receptor to a specific form and different forms, which can trigger different intracellular event after activation, what can be schematically shown here. And certain ligands of phenol selectively activates GS protein, and all these experiments shows that those GS selective ligands induce qualitatively different receptor conformations than others which are PTX sensitive, which means that they are GS not selective. Having this in mind, we decided to study a bigger cohort of these compounds in the same experiments without PTX and with PTX, and determine which of them are really selective to GS and which are not. And this is part of the list, and we found out that some of the ligands indeed are not sensitive to PTX, but some others, even if they are in RR stereo configurations, are sensitive to this. And based on these data and based on some molecular modeling, which I'm going to show in a minute, we made some kind of structure-activity relations to analysis to determine mechanism and determine differences in binding. So now I would like to show the molecular models of ligand-receptor interactions in the system. We are very happy that this is also a system which is studied by Brian Kobilka and Bob Lefkowitz, who got a Nobel Prize last year for inter alia, showing the molecular models of the receptor, molecular models in active form, in inactive form, and there is really quite a lot of different structures I could use in the modeling. So in those models, when we did the docking simulations, we found out that 4' hydroxy moiety, which seems to be the most important and decisive to determine whether the compound is GS selective or not, this moiety of the compound showing here interacts with either tyrosine 308, shown here, or lysine 305, shown here, depending on whether the model is in active form or inactive form. So that was the interaction we suspect that is important for observed activity. Now, what is happening with compounds which are not GS selective? And one of the most important in this analysis was a naphthyl phenolterol derivative, which does not have a 4' hydroxy or methoxy group here. It cannot interact with tyrosine 308, and we found out that location of this naphthyl moiety may be a bit different, interacting with and showing possibility and importance of interaction by pi-pi interactions with the receptor. This region of the receptor is rich in aromatic residues, and quite a lot of pi-pi interactions occurs here. When we did the RR4 methoxy 1-naphthyl phenolterol, which is also not GS selective, it has an oxygen here, but modeling shows that perhaps the pi-pi interactions are more important than creation of hydrogen bond with residues which are here, tyrosine 308 or lysine 305. So, based on these analyses and all these models and so on, we could divide the whole cohort of compounds into two groups with two different interactions patterns. And the group we call a group A, it has a hydrogen bond acceptor at the 4' position, and it does not have an after ring. These are the structures, and it means that oxygen or nitrogen in a 4' position should interact by a hydrogen bond with tyrosine 308 or lysine 305, and RR isomers of ligands from this group are GS selective. In a group B, the most important requirement is to possess a naphthyl ring. Whether it has an oxygen or in 4' position or not is really not important, and the predominant interactions are pi-pi interactions with a cluster of aromatic residues, and all of them, regardless of stereochemistry, are not GS selective. And to experimentally verify this type of hypothesis at the beginning, we decided to study binding affinity to the mutant, where tyrosine 308, this residue which is important to interact with a 4' oxygen or nitrogen at the molecule, is turned out and changed into alanine. And this is a comparison of binding affinities. This is a binding affinity of the compounds for Y-type beta-2 adrenergic receptor, and this is for the mutant. And as a control numbers, I also put the Ki values determined on beta-1 adrenergic receptor. This receptor also does not contain tyrosine in the position corresponding to 305 in that receptor. And as we can see, for group A, which we hypothesized that it creates a hydrogen bond with tyrosine 308 by the oxygen or nitrogen at the 4' position, we really can see that indeed binding affinity drops down. and this is shifted more to the values which we observed for beta 1 receptor. Here we have 0.35. Here it was 13.3, which is very close to the 14.8 observed for a beta 1 adrenergic receptor. And this trend is observed for all compounds from group A. Always the binding affinity for the mutant was shifted to the numbers which are closer to data determined for beta 1. Now in group B, which contains a naphthyl moiety, and we can see that mutation of this residue does not change binding affinity. Here it was 0.24, we still have 0.24. This is 0.34, we have a 1.9, so a small increase. Here is also numbers which are much closer to the wild-type data on beta 2 than to any numbers determined on beta 1. So I think this is a very elegant way to confirm the hypothesis we first draw based on structure-activity relationships, on molecular modeling, and so on, which eventually we could confirm that binding of that oxygen or nitrogen at the 4' position is really important for and binding to tyrosine 308 is really important for compounds in group A, which also implies that binding to this moiety is important for a receptor to be to be activated into GS selective form. Okay, and now I'll go very quickly through the conclusions. So I think I could convince you that stereochemistry of phenol and its derivatives is an important factor influencing a binding and functional activity of structures towards beta 2 receptor. RR isomers are the most active, whereas SS are the least active. Stereochemistry of ligands determine the mode of binding and a pattern of local interactions within the binding site, and we could evidence it by thermodynamics studies, but also stereochemistry of molecule affects the coupling properties of the receptor to different G proteins upon binding. And this is what I just have shown. Molecular modeling simulations of binding to the beta 2 receptor linked this difference between group A and group B with dichotomous interactions of derivatives with tyrosine 308 residue to beta 2 adrenergic receptor models. And indeed, studies on the mutated receptor confirm that hypothesis, and that GS selective derivatives they have significantly reduced affinity comparing to the beta 2 wild-type receptor. And obviously the ultimate goal of the project is to develop beta 2 agonists selective to specific receptor subtype, and in current understanding also specific to activated signaling pathway in the cell, in a body. And I would like to just quickly mention therapeutical perspectives. So 2 of the compounds which were developed in the project are tested in congestive heart failure, and as far as I know, there were short clinical trials to study those compounds in this treatment. And these are 4-methoxyfenoterol and RR4-methoxy1 naphthylfenoterol. This compound additionally has a cardioprotective properties. The compound which I also mentioned earlier, RR-ethylfenoterol, is very interesting for treatment of brain tumors because it does not causes significant cardiac problems, at least in the in vitro studies. now it's perhaps a time to test it in a more in vivo environment. Okay, and these are the acknowledgements, and obviously the central point in this project is Professor Irving Weiner and a couple of co-workers listed here from working for NIA. This is people from my university, and actually I should put another name, Artur Wnorowski, who is now working at the Earth's Laboratory on this project, and on also very interesting other aspects which I didn't have a chance to mention. And many part of experiments and synthesis of those compounds were done in SRI International under contract from National Institutes of Health, and my part of the research is also supported by Foundation for Polish Science in 2 different programs, founding programs. Okay, so that's it. Thank you very much, and again, congratulations of the award.
Are there any questions for our speaker? Mine is a very basic question. So the binding interactions that you showed between the naphtha moiety and the hydrogen bond, were they calculated by your system, or they were actually measured?
First, we calculated in the system. So, we were doing molecular models, and molecular models cannot tell you anything more than just allow you to make some hypothesis. This might be some possible mechanism which is different to another mechanism. So that was the first part. And then, as always, this is my lifetime idea. When you have a model, you then have to verify any results or any hypothesis with experiments. And I think this is the way we perform these experiments on the mutant. Very nicely confirmed hypotheses which were developed by molecular models first. You can calculate it, but this is only estimations in molecular models.
Very close.
Yes. Mm-hmm.
Very close. All right, great. Thank you.
Thank you.